Hello. Good afternoon, everyone. Thanks for joining. And I know you guys have a nice lunch, so this is a good time to take a short break and then come back. So hopefully you guys have enough caffeine in your system. All right. So today, what I wanted to talk to you about is how we in the company tackle some of the big major diseases from the lens of AI/ML, right? So in particularly, I will be talking about patient stratification of major depressive disorder using the data coming from UK Biobank. And I'll break it down in the next slides so that we follow this. All right. So just a disclosure that I'm a full-time employee of J&J. The opinions are expressed here are basically mine, not the company's. And I will not be discussing any particular products or any of the ongoing information regarding clinical trials provided by Janssen R&D or its affiliates. Right, so the agenda for today is that I will be breaking down the whole presentation into subsections. We'll start with the background and objectives about why depression is important and why do we want to study them. Then I'll give you a gentle introduction to UK Biobank. For those of you who have not heard about UK Biobank, it would be a good sort of summarization. And then we'll go over workflow and results. And then I'll follow up by the conclusion and what's the next coming in depression study. So let's start. All right, so why do we study depression? Depression is one of the major impacting disease that is impacting about 322 million individuals worldwide. And these are just the reported numbers. So in some countries, in Asian countries specifically, because I'm from Asia, I do know that depression, talking about depression is a stigma. So not many people seek medical attention or necessary treatment. So these are underreported numbers, but still 322 million individuals worldwide affected by depression. Very important to understand and study. One of the key challenges with MDD, major depressive disorder, is the patients are clinically and biologically heterogeneous. And that is one of the main reasons why it is complicated to identify a causal mechanism that can inform therapeutic development. This is one of the reasons why there is not a specific targeted drug out there to cure depression, because it's very, very challenging to identify the causal mechanism. So my team and I at Janssen, what we do is we basically break down this whole analysis, whole study into two objectives. First is, how do we leverage data-driven approaches to basically do the patient stratification of individuals so that we can better identify the patients who are having an MDD. And the idea here is to be able to leverage the electronic health records data, biomarker data, and cognitive measurements to basically tackle the stratification. So that's the first one. The second one is, once you have identified those subgroups or clusters, how do we now take a look at the underlying genetic architecture and try to identify what is the molecular or the causal mechanisms underlying the MDD within those subgroups, with the hypothesis that these subgroups or these clusters will eventually have different genetic architecture. So individuals belonging to one cluster may have different underlying genetic architecture from the others in the other group. So that's the overarching goal of the analysis of the study. Here, for the sake of timing, I'll be focusing on the first one. I will not be going over the genetic analysis component, because this is more catered towards the AI/ML. So I'll be discussing about the first objective, the impact. So one of the reasons why we want to study depression and using the data-driven approaches is that we would like to accelerate the enhanced patient target selection, drug target selection, as well as personalized treatment. So one of the reasons why we want to study depression is that depression has a clinically different, individuals with depression can have a different symptoms. So we would like to target, we would like to have the targeted or precise medicine catered towards those individuals. So precision medicine is very good, very huge initiative within Johnson & Johnson. So we would like to basically cater towards the precision medicine through the lens of AI/ML. All right. So for those of you who don't know what UK Biobank is The UK Biobank is a large population scale database of 500,000 individuals, which offers an exciting opportunity to examine patient stratification. Here I've just listed some of the basic data modalities that you will find in UK Biobank. So for example, you have a baseline measurement. So these are the information that individuals, when they go visit a UK Biobank registration center, they will collect information about their overall health status. So these are basically embedded in the questionnaires. Physical measurements like ECG, blood pressure, heart rate, and then you'll have the cognitive assessment measures. So these are all these very, very baseline measurements that have been collected for individuals. And these are collected over a period of time as well. So you have that longitudinal aspect if you are really interested in study how disease progresses over time. Then you have some enhanced measures. So these are some of the specific biomarkers, accelerometer data, imaging data that you have within UK Biobank. You have also the longitudinal EHR data available for these 500,000 individuals. Then you have the logs of their cancer and death registries. And then you have the plethora of genomic data, which includes whole exome sequencing data, SNP array data, and whole genome sequencing data. And this is, again, an ongoing data repository, so new data is going to be added because UK Biobank have collected the data and they keep on increasing the different data modalities. So there might be more modality of other omics data that may be coming soon to the UK Biobank. So here I just wanted to give you a very high level overview of what UK Biobank has to offer. So this is the very source of the data that I'm going to be using. All right. Next, so once I know where my data is coming from, the next thing is I wanted to know what set of patients, what are the individuals, what is my cohort. So what we've done is we've actually identified about 60,813 cases of probable MDD individuals in the UK Biobank, and about 231,787 controls or normal individuals for this analysis. And the way we've selected our cases for probable MDD is based on some selection criteria based on their responses to mental health questionnaire and their digital clinical diagnosis that's embedded within the UK Biobank. So from when we do these cohort selection, we basically get some of the basic statistics or some of the basic metadata about their gender. So we do know from literature that more female tend to have a depression, and that's actually apparent in the UK Biobank data as well. You can see here in the distribution of male and female, more female are diagnosed with depression. And also the second plot that you see at the bottom right corner is the age distribution of your cases and control. So here you can also see the age. This is the age at the time when they visit the recruitment center. This is not the age of onset for depression. Just wanted to clarify that here. And overall, we observed about 21% prevalence of probable MDD in the UK Biobank. All right. So moving next, now that we have a clear understanding of what cohort, what's the sample size, wanted to give you a workflow overview. So we started with the data. So from UK Biobank, you've seen all the data types. Now I wanted to focus on specific data that I've utilized for this analysis, for this research question. So I'm using blood and urine biomarker data. Then we've collected the cognitive function measures. These are the cognitive function measures that have been taken on individuals so that we know their cognitive ability. And then you have the clinical phenotypes. This is basically to capture the comorbidities amongst those patients and see if we can find any more insights. So this is the data or / features that we'll be using. Next is the feature selection step. So now that we have the data, what we wanted to do is we have a definition of a case and a control. What we wanted to do was we wanted to use machine learning algorithm to classify cases from control when we do that, what we do is we actually also learn about some of the important features that distinguishes your cases from your control. So feature selection was done using exactly this very basic technique. And once we identify what are some of the clinically meaningful features, we basically brought those features forward for clustering analysis. So what we did was we take a look at, for example, top 50 clinically meaningful features. We basically use autoencoder to basically narrow down and to basically understand the nonlinear relationship between these features. And then we use the actually the middle layer, which is the most compressed and abstract layer of autoencoder, to basically as an input for the k-means clustering. So we use this basically k-means clustering algorithm with autoencoder for identifying clusters of individuals with probable MDD. So this is, again, a very high-level overview of our methodology here. All right. Next, I'll dive into some of the results, technical results. So for feature selection, we built a XGBoost model. We used the balanced data set to evaluate the model, as we know that there is a class imbalance here, 60,000 cases and 230,000 controls. The data was split into 70% training, 20% test, and 10% validation. And for the predictive performance, we have a separate set of 20% test set, and our predictive performance was measured at 72% on the test set, which is pretty consistent with what others have published in the literature. So this is also one of those sanity checks that you do and see that whether the results are actually in line with what is already published. And here I'm just showing you the evaluation of the training model in the AUC PR curve, basically, which is 73% overall. So once we build the classification model, next I wanted to show you is the clinically meaningful features. So now that we are in the era of ethical AI, explainable AI, the idea here is to be able to explain what basically how the decision was being made, right? So basically here in this plot for all the data scientists who are here sitting here will recognize this is a SHAP summary plot. For those of you who do not understand this plot, I can just quickly break it down. So on the y-axis, you have the most important features, the top feature being the most important one. The x-axis shows you the logs odd ratio of someone having an MDD with that feature. So the x-axis is just, again, log odds ratio of having an MDD. The colors here denote something. The color here represents value. So when you have red color, meaning the feature value is high. Blue color, meaning the feature value is low. So if you take a look at the very first feature, which is testosterone, what that particular plot tells you is that the lower the value of testosterone, which is indicated by the blue color, the higher the log odds of having an MDD. And this is very consistent. This is consistent with what we have seen in the literature, meaning that more female have depression. So not only you can see with explainable AI, we not only see the feature importance, but you can also see the directionality of effect of how each feature is contributing towards the overall likelihood of someone having an MDD or depression. So again, this is just an example of clinically meaningful features we have. On the right side, I just wanted to summarize highly overall what are the important features that we've seen. So we've seen some of the important biomarkers like testosterone, varying levels of c-reactive proteins, vitamin D, and then we've seen for the clinical comorbidity clusters, we've seen our features, we've seen mental and anxiety disorders, substance usage, and suicidal attempts. So these are normally some of the features that you would normally associate with depressed individuals. All right. So once we identify our clinically meaningful features, next we wanted to do was basically do the clustering. So here I'm going to show you the overall clustering results. So we identified about 8 unique clusters. Now we started with data, feature selection, now the clusters, right? So we've identified 8 unique clusters. These clusters are driven by depressive symptoms, behavior and biomarkers, and actually also are enriched with clinical other comorbidities. And I'll break it down in the next slide. But what we basically were able to identify is that these cluster profiling that we did, that I'm going to show you in the next slide, further validates that our subtypes and feature dominance is actually consistent with the literature, which is something very, very, very encouraging to see as a data scientist, that your results are in line with what others have published in a clinically meaningful way as well, not just technically in terms of what are the accuracy or what are the accuracy of the model, but also clinically it makes sense. This is one of the reasons why we would like to do AI/ML in healthcare in generally, right? We would like to see clinical significance. Here on the plot here on the bottom, I'm just showing you on the x-axis, you see some of the informative features from the phi codes, meaning the clinical diagnosis, and the markers are the biomarkers on the x-axis. And on the y-axis, you see the standardized mean. So basically the values of each features break it down by different clusters. So different line represents different clusters. So here you can see how each feature was contributing to different clusters and which clusters are dominated by which features. So this is again a high-level overview of what distribution have for all these features. And on the right side, you have the t-SNE plot showing you the overall cluster space with our 8 clusters. All right. Next here, now that we have the cluster, I wanted to further deep dive into specific clusters and see if it makes sense, right? So here I'm showing you a result of cluster number 1, which is actually driven by disorder of lipid metabolism, mental disorder, headache, high levels of c-reactive protein, glucose, triglyceride, urate, and then lower levels of vitamin D. So here on the x-axis, you can see the distribution of all the phi codes, all the clinical subtypes, or all the clinical features. And on the right side, you see all the biomarkers, the blood biomarkers. So this way, basically, when you break it down to and profile the clusters here, you can see how each feature contributes to the strata, to the group. All right. I have one more example, which is very exciting to see. So this is profiling of cluster number 7, which revealed that we may have a potentially severe MDD subtype, which is driven by substance abuse, suicidal ideation attempts, as you can see it from the line plot. That on the left side, you see that there is a peak of suicidal ideation and attempts, substance abuse, and poisoning by the psychotropic agent, which is probably overdose, drug overdose. And on the right side, you see that individuals in this category are taking more reaction time, meaning that they have less cognitive ability than the normal normal individuals, and lower levels of vitamin D, which is consistent with individuals who are depressed because they don't move a lot. So their vitamin D levels are actually lower. So this is again representing a severe MDD phenotype that we observed in one of the clusters. And here I'm breaking down the cluster metadata so they can see basically the mean age distribution amongst this cluster is about 55 years old, 55 years. Males to female ratio is about 1 to 1.25. Percentage of how many individuals are clinically diagnosed in this group, 85%. And this is again, we never feed this information a priori. This is again, something that we observed. All right, next. I just wanted to conclude what we've seen here today. So the machine learning algorithm that we developed was able to predict the probable MDD from cases from control with about 72% performance. The top features that we basically selected were actually able to help us identify some of the informative blood biomarkers and also clinically meaningful phi codes or phenotypes. We were able to identify about 8 unique clusters. And some of the clusters, for example, cluster number 1 showed that it is driven by mental disorders, higher level of blood serum biomarkers, and lower levels of vitamin D. Cluster number 7 was driven by, like I showed you in the previous slide, it showed a more severe phenotype of MDD or subtype. And overall, basically, the approach that we've built here, it could be disease agnostic. It could be applied to any of these diseases for any disorder to improve the patient stratification. Ultimately, our goal is to be able to boost target identification and validation effort at Janssen. So in terms of feature direction, what do we do with this? Where do we go from here, right? So the feature is basically we would like to replicate this. Just because we've seen it in a data set, that doesn't mean that it's consistent. So we would like to assess our stability and validate that through independent cohort. So when you see a subtype in one population, do you see that in the second population? If you see that, then you know that your results make sense. And also we'd like to integrate other types of data modalities in UK Biobank, for example, neuroimaging data, as well as actigraphy data. So with that, I would like to thank the team at Janssen, specifically our colleagues, my colleagues Dong Nu Trong and Katya Kramatsova, who actually worked with me on this project and are leading the genetic analysis after the subtypes have been identified to basically accelerate our drug discovery portfolio. And also would like to thank the Johnson & Johnson leadership team, who have been very supportive and in terms of encouraging me to come to present. And yeah, overall, it's a great collaboration with UK Biobank. And yeah, that's all. Any questions? This is not available publicly, not at this time, because pharma companies have signed an exclusivity period, which means that because they've invested millions and millions of dollars in UK Biobank, that's how UK Biobank got their funding. So they've given them at least 9 months to 12 months of exclusivity period. Every single time a new data comes in, actually the pharma companies who have invested gets 9 to 12 month exclusivity period. After that, it will become public. Sorry?
So what is the due on this?
So it really depends on what data type that you want to take a look at, right? So for example, if you want to take a look at the neuroimaging data, then maybe the exclusivity period is even beyond 12 months. Maybe it's 16 months. So it really depends on what type of data that you are looking at. But right now, I do know that it's at the hand of pharma companies. It's not public, but eventually it will become public. Yeah.
Based on the subtypes, right, and based on the clinical research out there,
Yeah.
are there any defined subtypes that they've identified when they're researching after? You did mention that some of the results that you observed
Yeah.
sort of align
Yeah.
with what you want to see. So this is sort of curious about how many subtypes that you identify sort of align with what the clinical
Yeah.
research is.
Yeah, so that's a great question. So what we've identified is not specifically a subtype, but the features that are driving the subtype are consistent. And here the underlying goal is really we wanted to get through the target discovery. So what we wanted to do is if you have two groups of clusters, two different groups of individuals or patients, what are their underlying genetic architecture? They may have different or similar symptoms in the same cluster, but if they have a similar genetic architecture, then this is what we want to get at. So our hypothesis was to begin with, different clusters have different genetic architecture so that we don't find one medication that would be cater for all because it may not work against one group. So that was the idea behind this.